We're gonna be looking at DNA as the code of life. That's your first topic for the year. DNA as the code of life. But before we get into DNA, I would like us to do a bit of a revision from grade 9 and 10 work on the cell. Obviously, this would be your animal cell as it has no chloroplast and so forth, right? Good. Let's do a quick recap on the cell. It will help us understand this topic of DNA. We all know that on the outside of the cell, the border control of the cell, basically what controls what goes in and out of the cell is your cell membrane, right? It controls what goes in and out of the cell. That's your cell membrane. And then inside your cell membrane, you will have your cytoplasm, your whole entire cytoplasm. Let's do this. Good. Your whole entire cytoplasm, right? Good. Where all the organelles of the cell are, and basically where all the reactions of the cell take place. And then you'll have other organelles inside the cell, for example, such as your Golgi body. That's the Golgi body as part of your cell, right? Good. We don't have to go into detail as what does the Golgi body do. We're more interested in other organelles inside your cell. And then you have the endoplasmic reticulum, which is divided into the smooth and the rough endoplasmic reticulum. The rough endoplasmic reticulum is rough because it has some ribosomes attached to it, right? Great. And since this is the animal cell, you'll see your centrioles or centrosomes right there. And then one of the most important organelles that we must know for now is your mitochondrion. Mitochondrion. We all know that the mitochondrion is responsible for providing energy through the process of cellular respiration. Basically, it's the ESCOM of that cell. Now, what we need, I see all of these, what we need is this organelle right here. In order for us to understand DNA, we need this organelle right here. It is called the nucleus, right? We need the nucleus. Now let's take a quick recap on the nucleus. The nucleus is made up of a double membrane, right? A double membrane with what that we call the nuclear membrane, right? The nuclear membrane. And the nuclear membrane, if you look at it properly, the nuclear membrane has got pores on it. These little pores we call nuclear pores. They're like little holes. Pores are like little holes, similar to the pores you have on your skin. The nuclear membrane has got pores. What are they there for? To facilitate the transport in and out of things such as proteins and RNA. And then this thread-like structure that you see inside the nucleus, basically it is your chromatin network. Chromatin network. We learned in grade 10 that the chromatin network basically is unorganized DNA. And then once it winds up basically to form that little structure that looks like this. This looks like a ears. That will be your chromosome. So it will wind up, stop being thread-like, it will wind up around the histones and form what we call your DNA. So this is one and the same thing, just that one is coiled up or wind up, and then the other is just loose around the nucleus. Then inside the nucleus, right, you will have the following, similar to the cytoplasm. Inside the nucleus, you will have your nuclear plasm. The nuclear plasm is your jelly-like substance, similar to the cytoplasm. It's not really the cytoplasm. And right on the inside of the nucleus, we find the nucleolus, where you find RNA, ribonucleic acid. That is your nucleolus. Great. Now that we have revised the cell, more particularly, I wanted us to revise the nucleus. So we can go back and start this great topic of us, which is DNA. I love it, love it, I love it a lot. Great, let me get rid of this. So, what are we going to be doing when we look at DNA? When we look at DNA as the code of life. So, DNA is the code of life. So, let's quickly do a summary. First things first, when we look at DNA, we're gonna look at what is DNA. Basically, this is a summary of what we're gonna do. What is DNA? And then you'll find out that DNA is a nucleic acid, and there's two types, right? There's DNA, the actual DNA, and one is called RNA. And then we're gonna look at the structure of DNA, structure, and then the history. We cannot look at the structure without actually going into the history to see who actually came up with the, or discovered, not came up with it per se, but discovered the structure of DNA. That's when we'll be talking about Rosalind Franklin, Maurice Wilkins, Waston and Crick. Then location, GPS. Where do we find DNA? Where do we find it in the cell? Then, after we look at where do we find it, we're going to be looking at the role of DNA. DNA. What is the function of DNA? Why do we have DNA? For example, we're going to be looking at heredity. For example, that's one of the things. What is heredity? Heredity is basically passing on genes from the parent to the offspring. This year we must be very careful to use the word offspring, not children. Come on, we're no longer children in grade 10 and 11. We use the more nicer words such as offspring, right? Good. Now that we are there, let's dive into it. Let me dive into those quick notes of mine. Great. So this is part of your paper 2, life sciences paper 2, DNA is the code of life. This is what we did. Basically, you can go through this to remind yourself of different parts of the cell so that when we go through it, you know what we are doing, and basically do this activity while you're at it to see that you completely remember most of the parts of the cell. What we are interested in now is the following, right? There are certain terms that you must be very wary of because they might confuse you. They sound the same, but they're not the same. Words such as nucleolus, nucleoplasm, cytoplasm, and ribosomes, especially the first three. The cytoplasm, for example, is part of the protoplasm outside the nucleus. Basically, it's everything except the nucleus inside the cell. The nucleoplasm is part of the protoplasm within the nucleus, that jelly-like substance in the nucleus. Then the nucleolus is the structure in the nucleus, the central one that I showed you, responsible for forming ribosomal RNA. Then other words of terms include chromatin, chromatid, centromere, chromosome, and chromatin network. You must learn how to differentiate these words. For example, chromatin is the DNA containing network found in cells in interphase, basically the non-dividing phase of the cell when the cell is basically growing. Yeah? Good. When the DNA is unwanted, the unorganized DNA. Chromatid is the individual threads from a chromosome, right? For example, remember we did this. If we have this, this is a chromosome, and these are two sister chromatids. These are sister chromatids. Good. Then the centromere is the structure that holds two chromatids together. Basically, is this one. This is the centromere. Basically, remember in mitosis, that's where the two, spindle fiber attach during metaphase. Good. The metaphase plate. Then chromosome is a thread-like structure made up of DNA that carries hereditary information. Basically, it will look like disorganized DNA. And chromatin network, visible thread-like structures in the nucleus of an inactive cell. Great. Now, let's look at the structure of DNA. In looking at the structure of the DNA, we're gonna start with the history. Who came up with the structure? I don't know, but you know, well, it's not really rumor. History has it that Rosalind Franklin was actually one of the pioneers in finding the structure of DNA. X-ray she used X-ray to actually visualize DNA, the structure of DNA, the first structures. She had an image. There's a famous image of it. And then she worked hand in hand in a university with Maurice Wilkins. They researched together on the structure of DNA. Obviously, women in that era were not well sort of when it comes to uh, science, right? Other guys that were busy with DNA concurrently were Waston and Crick. Now, apparently, what happened was that the X-ray image found by Rosalind Franklin in 1952 was taken by Maurice Wilkins and went to these guys and said, well, is this similar to what you're working with? And then they used that image. They were like, okay, yes, it is similar. And then they came up and with the structure of DNA. They call it they came up with the 3D double helix model. So the shape of DNA is known as the double helix. And then after that, years later, well, that was after Rosalind Franklin died. Watson and Crick received the Nobel Peace Prize for not the Nobel Peace Prize, the Nobel Prize for the discovery of the structure of DNA. Maurice received an award for his X-ray photography. It wasn't really his. I am judging him. I'm judging him hard. That was in 1652 when they received the Nobel Prize. But I, I personally think Rosalind Franklin, as a woman, is the mother of discovering the double helix. If she didn't die, she would have also came up to the same conclusion as Waston and Crick. So basically, what is the double helix? This is the double helix structure, right? Good. Now, just like each and every organic compound, remember we said that DNA is an organic, hey, what am I doing? Organic compound. Now, organic compounds are made up of monomers. What are monomers? Monomers are building blocks. You need to follow me very carefully. Remember in grade 10 we said we have things such as nucleotides. So nucleic acids, sorry. Nucleic acids. What are nucleic acids? Nucleic acids are things such as DNA, or organic compounds such as DNA and RNA. But what makes this DNA and RNA? What are their monomers? Their monomers are basically something called nucleotides. So that is basically what we're going to be looking at when we look at the structure of DNA. So nucleic acid is an organic compound. It is made up of building blocks called, or monomers called, nucleotides, and these form both RNA and DNA, as they are both what? They are both nucleic acids. They're made up of monomers called nucleic acids, right? We have already said nucleic acids, there's two of them, deoxyribonucleic acid and ribonucleic acid. The reason that they differ is that they differ in structure and name, precisely the name, because DNA, somewhere on the pentose sugar, it's missing an oxygen. It's missing an oxygen. And then it's just the hydrogen. And then thereby RNA, somewhere on the pentose sugar, there's OH, it has an oxygen, so it is ribonucleic oxygen. So this one is deoxy, without oxygen, ribonucleic acid. That's why they're named differently. But there's a lot of difference in terms of their structures, as you'll see later. One is a double helix, meaning there's two strands, and then the other is basically a one strand, a single strand with a difference in one nucleotide. But anyway, so what is basically what is the nucleotide? This nucleotide that we speak of. When we say a nucleotide, your nucleotide must be made of three things, ne? Smaller little things. This is the phosphate group. The phosphate group will be connected to the pentose sugar, the pentose sugar molecule over here. Together, these two make the backbone. These are the backbones of DNA and RNA. And then attached to the sugar again is the nitrogenous base. We have four types of nitrogenous bases: adenine, thymine, guanine, and cytosine. Right? Great, but we'll get to that later. Now, where is the location? GPS. GPS location, sila. Where do we find DNA? DNA is found mainly, we said, in the nucleus, right? That is, so we give it a surname. That is nuclear DNA, right? Nuclear DNA inside the nucleus. Now, the DNA that is found outside the nucleus, that is extra DNA. We call it extra nuclear DNA. That's DNA found outside the nucleus. And then what do we call it? Where do we find it? We find it in chloroplast. Obviously, in animal cells, you don't have a chloroplast, right? Good. So you'll find it in plants or some protists. For example, some protists have a chloroplast. So that means they will have a chloroplastic DNA. So that will be your chloroplastic DNA. These are all part of extranuclear DNA. And then mitochondrial DNA. So all animal cells will have this mitochondrial DNA, but they will lack chloroplastic DNA for the simple fact that animal cells do not have a chloroplast. I've never seen a green human being, and the whole does not count. Right. Back to the structure of DNA. So we said it is made up of a sugar, a deoxyribose sugar, meaning it lacks an oxygen, a phosphate molecule, and four types of nitrogenous bases. Adenine, denoted with a capital letter A, thymine, represented by capital or denoted by capital letter T. And then adenine and thymine, we call them base pairs. They are known as base pairs, meaning they will always bond with each other. Basically, these two are married. So for slatlane type of thing, right? Good. Wherever you see adenine, wherever you see A, that means that A will always bond with T, like here as well. Wherever you see T, like here, that means there will always be A. So whenever you see the one, the other one, the partner follows. Similarly, cytosine and guanine are also your base pairs. They are also a couple there. They are married for life. Basically, the base pairs. Whenever you see guanine, you're going to see cytosine. It's going to be paired with cytosine. Whenever you see cytosine, it's always going to be paired with guanine. Good. And how are they paired? They are paired using some weak hydrogen bonds. So they use hydrogen bonds. So whenever they ask you in an exam, the bonds formed, they are weak hydrogen bonds. Basically, these bonds are broken during DNA replication. And then once they bond, these base pairs, they bond together all in all to form this double helix structure. Double helix, if you wanna imagine it, just imagine your ladder, step ladder. You see when in our languages we call it step ladder, properly a step ladder. So the step ladder, you take it, you imagine if you could have the strength to rotate it. That's basically how your DNA double helix would look like. So what you need to remember, your nucleotides contain a deoxyribose sugar for DNA. That's specifically for DNA. A phosphate molecule, and then attached to the sugar will be one of the four nitrogenous bases. The nitrogenous bases: adenine, thymine, cytosine, and guanine. Yeah? Good. Now, what you need to notice, let's have a look at the structure very quickly before we explain histones. This over here is one nucleotide, and this over here is one nucleotide. Your nucleotide, not nucleotide, yeah, it is a nucleotide. Ne? It's one nucleotide. The phosphate, the sugar, and the nitrogenous base ne, forms one nucleotide. So if we were to count how many nucleotides we have here, this would be 2, 4, 6, 8, 10. We have 10 nucleotides all in all. I hope we get it. So for your DNA, basically to wrap when it winds up, it is wrapped around these little proteins called histones. Those are the proteins whereby your DNA wraps around. Good. Lastly, what is the role of DNA? Most DNA doesn't code for anything. Basically, if you have a long chromatin network, just to give you an example, long chromatin network like this, only a certain portion of it will basically code. For example, let's say, why am I erasing here? Where's my highlighter? Great, I don't need yellow. Yeah, let's do this. You'll find that the coding region for your DNA is this one. Just to make an example, this is the coding region of your DNA. Good. Then the rest does not code. It's still DNA, but it doesn't code for the genes and proteins needed in your body. So we call it non-coding DNA. So most of it in your body is exactly like that. It's called non-coding DNA. And then the one that codes is called your coding DNA. Basically, it contains the code. Remember, you'll see later on we have those nitrogenous bases, for example, C, cytosine, thymine, and adenine. Three of them, they are called a codon, meaning they will code for a specific amino acid, like arginine, and and different types of codons would code for different types of proteins. So those proteins will line up, basically will bond. They will bond together to form a protein. Because remember in grade 10, we mentioned that all of these amino acids are building blocks or monomers of proteins. So that's what we mean when we say it's a coding DNA. So what are the main functions of DNA? It controls all functions of the cell. That's why we say nucleus controls all functions of the cell. It is due to DNA. Then to regulate the function of genes and then pass on hereditary information from parent to offspring. So you have that big nose because of your DNA. It goes in line with your whole family from one person to the other. So that this will basically conclude our lesson on DNA for now. So let's see. Did we cover all that we said we would cover? Did we looked at the what is DNA, the structure, and the history. The structure is a double helix structure made up of nucleotides. Nucleotides are made up of what? You can do look at this mind map. We have nucleotides, and nucleotides are made up of three things: your phosphate, your sugar, and your nitrogenous bases, right? And the history behind it is that Rosalind Franklin and Wilkins worked on it, and then Waston and Crick in the year 1953 discovered the double helix. Okay, yeah, discovered the double helix model or structure of DNA, right? Then location, it is found in three places. It is found mainly in the nucleus, that's for every cell, in the mitochondrion, and then in the chloroplast. The DNA found in the chloroplast and mitochondrion is called extranuclear DNA, and the one found in the nucleus is nuclear DNA. Then the role of we just did the role of DNA. I hope you